Hey guys, welcome back to my channel MedEdophilia. In my previous video, I had discussed about the nose and the tongue. And in today's video, I'll be discussing about the most interesting sense organ, which is the eye. And I would say this is the most important topic among all the sense organs from NEET exam point of view, followed by the ear, which I'll be taking in the next video. Firstly, let's observe the picture which shows the eye externally, and let's try to describe its part. The most obvious structures we can point out is the eyelid and the eyelashes, and then just based on our visual examination we can differentiate the eye into the white part and the colored part. The white part known as the sclera and the colored part. For most of the people it's black, but it's not necessarily black for all the people, as we know some people have blue eyes and so on. So the part which is responsible for the color of the eye is the iris. You can see that the iris has a central aperture known as the pupil. Then to the tip of one end we can see a small gland, actually it's present little towards the inner side, which is known as the lacrimal gland, which is responsible for the production of tears from the eye. These tears keep our eyes moist and provides a flushing activity and helps to clean the eye surface. The next part is the nictitating membrane, which is also called by some people as the third eyelid. Nictitating membrane is vestigial. Its main function is protection. The eye is lot more from what we see externally. So let's quickly dive into its anatomical features. And before that, just a small mention, as we all know eye is one pair and is nearly spherical in shape in most of the adults, and it is located in sockets of the skull called orbits. Let me draw for you so that you can understand. Okay, so going into the anatomy of the eye, firstly the wall of the eyeball is composed of three layers S, C, R. The externalmost layer is the sclera, and the middle one is choroid, and the innermost layer is called the retina. So from this part every description, every word is very important about each layer of the wall of the eyeball. So please listen carefully. Sclera is the externalmost layer, which I already mentioned. It is composed of the dense fibrous connective tissue, and the sclera helps in maintaining the shape of the eye. So the sclera, which is also known as the white of the eye, is the externalmost layer of the eyeball, and it is composed of the dense fibrous connective tissue, and it helps in maintaining the shape of the eye. The anterior portion of the sclera is known as the cornea. Cornea is avascular. So what is avascular? A means no. So avascular or non-vascular, and it is composed of non-keratinized squamous epithelium. So sclera is made up of dense fibrous connective tissue, whereas cornea, which is the anterior portion of sclera, is made up of non-keratinized squamous epithelium. Sclera was the white of the eye, but cornea is transparent. The anterior portion of sclera is transparent. So since cornea is avascular, cornea is the most commonly transplanted part of the human body, and it is the most accepted body part, that is graft or transplant. So here you can expect two questions. One, which is the most commonly transplanted body part, and the second one, the reasoning why it is the most accepted graft used for transplantation. So this is it about the external layer. Now moving on to the middle layer, which is the choroid. So choroid looks bluish in color because it contains many blood vessels. So like how we saw the differentiation of the external layer in its anterior part and the remaining part as sclera and cornea, here too we have a differentiation. Now the choroid layer is thin over its posterior two-thirds, and The remaining one third, the anterior part, is thick, and this thick part of the anterior region is called the ciliary body. So I repeat, the choroid layer is differentiated into the posterior region and the anterior region. The posterior two-thirds of the eyeball is thin. So thin posterior for two-thirds and thick in the anterior, forming the ciliary body. So here I will tell you a short mnemonic or a trick to remember this, because sometimes when you see these statements in your MCQs, this might become little confusing whether it's thick in the posterior or anterior. So here's it. Auntie's body is thick, okay, or fat. So auntie's body is thick. So auntie for anterior, body for ciliary body, and thick. So you can remember it in any way. Auntie's body is fat or thick, anyway, just for you to remember. Okay, so now the ciliary body, it continues forward to form the iris. So this iris is responsible for the color of the eye, which is different in people. Blue, black, brown colored eyes. This iris is pigmented and it is opaque, and the common name of iris is the diaphragm of camera, as its muscles help in regulating the diameter of the pupil. So many people get confused between the iris and pupil, confused between its functions also. So you have to remember that pupil is an aperture, okay, and iris surrounds the aperture called the pupil. So pupil is an aperture and iris is surrounding the aperture. The muscles of the iris help in controlling the diameter of the pupil. So when we enter a dark room, the pupil dilates or expands. And when we enter a bright room, sudden bright room, the pupil becomes narrow. It constricts, we say. So dilates in dark, DD, dilation in the dark conditions. Pupil constriction is mediated by the parasympathetic nervous system, PANS, and pupil dilation is mediated by the SANS, sympathetic autonomic nervous system. So the next structure is the ciliary muscles. Ciliary muscles are circular muscles which is just present as a wrap around the lens. The main function of the ciliary muscle is to alter the shape of the lens. Here, by altering the shape, the focal length of the lens changes, and thereby the power of accommodation of the eye changes. So when focal length and power are inversely related to each other, right? So since the focal length of the lens is changing, the power is also changing. So what do you mean by power of accommodation? So it is the unique ability of our eye to see very very far objects and very near objects. So power of accommodation is the ability of the eye to change its focus from distant to near objects and vice versa. This process is achieved only by the lens changing its shape. So now let's talk about the lens. Lens is a transparent crystalline structure. So it is held in one position by the suspensory ligaments, which is attached to the ciliary body, and it is avascular. So in old people, this crystalline nature of the lens, the transparent nature of the lens, gets lost and the lens becomes opaque. This leads to a condition known as the cataract, mostly observed in old aged people. So the ciliary body, can you recollect what it is? We learned three terms: ciliary body, ciliary muscles, and we are also going to learn one more term, which is ciliary process. These terms will be confusing for you. Let me explain it one by one. So what is ciliary body? So auntie's body is fat. Yes. So ciliary body is the thick thick portion of the choroid in the anterior region, and it is the ciliary body which continues forward to form the iris. And the next one is ciliary muscle, which we saw it is wrapping around the lens, which helps in altering the shape of the lens. Next one is the ciliary process, which is extensions of the ciliary body, and the ciliary process help in the secretion of aqueous humor. The aqueous humor is the thin watery fluid which is secreted every day by the ciliary process. So these three terms, ciliary body, ciliary muscle and ciliary process, you have to be really careful with these terms. These are going to trick you so much while solving MCQs. So make sure you practice them well. And there's one more term I told you, which is suspensory ligaments, which helps to hold the lens in a position. So what happens when we see a near object? The ciliary muscles contract, the ligaments relax, the suspensory ligaments relax, and the lens becomes thick. It bulges. So its radius of curvature decreases. So from this part you will get questions like this. So like what will happen to the ciliary muscles, what will happen to your suspensory ligaments, and what will happen to the shape of the lens when you focus a near object and when you see a distant object. So if you're not able to visualize it, I am here with another mnemonic, which is NCERT, the book, the NCERT book, but the E is absent. You just need to keep that in mind. N for near object, C for ciliary muscles contract, both C, ciliary muscle and contract, and the R for relax, the ligaments relax, and then T for thick, the lens becomes thick and bulges. So with this you can replicate for what happens when you see a distant object. So this is an amazing trick which will help you save a lot of time in answering such type of questions framed on the same line. So okay, so till now we only saw about the different structures and its functions, and you wouldn't have been that much clear and you wouldn't have been able to visualize all the structures properly. So let's get back to our picture. Let me draw it here so that you can understand each picture, the structure of each and every part which I discussed now. So this is the lens. Remember the human lens? So you tell me in the comments. It's quite easy and I hope you all must have known by now. Is it convex or concave? So comment below. The human eye lens is biconvex or biconcave. Okay, so this is the human lens, and here is the ciliary muscle wrapped around the human lens, eye lens, and this is the ciliary body which has the ciliary process, and it is attached to the lens by means of suspensory ligaments which helps in holding the lens in the position. Now let's go to the image. This will be like a revision for you. So each time I point out the structure, try to recollect the functions you have learned till now. So now the externalmost part of the eye, which is the white of the eye, as you can see, is the sclera. And then the anterior part here you can see, which is transparent. Notice this is not white in color, this is transparent, and it is called the cornea. Then coming to the middle layer, which is the choroid. Can you notice the thickness in the anterior and the posterior part? Okay, so it has, that is the anterior part has the ciliary body which continues forward to form the iris. The iris has a central aperture. Iris surrounds the central aperture known as the pupil. Then here is the lens. Okay, so here you can see a structure which is labeled as the conjunctiva, right? So this conjunctiva is the inner membrane or folding of the eyelid, which is highly vascular. This conjunctiva covers the white of the eye, and so it is technically the outermost. And this conjunctiva is made up of non-keratinized stratified squamous epithelium, just like the cornea, which is also made up of non-keratinized stratified squamous epithelium. So now the space you can see here, can you see there is a space between the cornea and the lens. This space is known as the aqueous chamber, and that space which is between the lens and the retina. What is retina? It is the innermost layer of the eyeball wall. Okay, so the space between the lens and the retina is the vitreous chamber. So now let's see the difference between the aqueous chamber and the vitreous chamber. The aqueous chamber is enclosed or filled with a fluid known as the aqueous humor. Recollect, the aqueous humor is a thin watery fluid. So what is the function of aqueous humor? Aqueous humor provides nourishment and oxygen. And who secretes aqueous humor? It is the ciliary process. Yes, it is secreted every day by the ciliary process. Okay. The second one is the vitreous chamber, which is filled with a transparent gel. Here you can see the aqueous humor is thin and watery. Now here it is a transparent gel, which means the thickness of aqueous humor and vitreous humor are not the same. Vitreous humor is thick, it's a gel. Okay, getting the difference? Okay, so vitreous humor is a transparent gel. So this vitreous humor is provides interocular pressure, that is the pressure of the eyeball, and it is responsible for the shape of the eyeball. And when is it secreted? It is secreted during the embryonal stage. By whom? By the retina. So the vitreous humor is a transparent gel. It is not thin and watery like the aqueous humor. It is responsible for the shape and interocular pressure of the eyeball, and it is secreted only during the embryonal stage, not like aqueous humor which is secreted every day. Okay, and it is secreted by the retina. So here's a homework question for you. What is the difference between this aqueous humor and the tears? Is there any difference or both are the same? Are they of the same composition? So I want you to think on it and comment your answer below. So here let me draw. There is a canal known as the Canal of Schlemm. Canal of Schlemm responsible for the drainage of aqueous humor. That is, it helps in the draining out of excess of aqueous humor. So when this canal of Schlemm is blocked, it leads to blindness. This disorder is known as glaucoma, or commonly referred as kalamotia. So the blockage leads to blindness, and this is mainly due to the increase in interocular pressure due to excess aqueous humor and not getting drained properly. And apart from the aqueous chamber and vitreous chamber, there are two other chambers which you find in some of the books, which is the anterior chamber and the posterior chamber. Anterior chamber is between the cornea and the iris, and the posterior chamber is the space between the iris and the lens. So here you can see in this picture, anterior and posterior chambers are not the same as aqueous chamber and vitreous chamber respectively. So know the difference and know where the space is enclosed between. Okay. Next we move on to the retina, which is like super important for NEET point of view. So if you understand this, even the mechanism of vision will be easy for you. So pay attention. So the retina is the innermost layer, and it is known as the neural tunic because it is composed of three layers of neural cells. The innermost is the ganglion, followed by bipolar cells, and the outermost is the photoreceptor cells. If you're finding it hard to remember, just remember it in this way: gang of lion surrounded by or followed by two polar bears taking a photo. So, what is it? Gang of lion followed by two polar bears taking a photo. So this point you need to keep in mind whether it's inside to outside or outside to inside. So the gang is inside. The gang of lion is inside. Ganglion. Ganglion cells are inside. Okay, and the outermost is the photoreceptor cells. So this inside to outside is the anatomical basis. Okay, so based on anatomy and histology, the cells which are located innermost is ganglion, next is bipolar, next is photoreceptor. Same for light transmission path. First it is the ganglion, and next is the bipolar, next it is the photoreceptor cell. But the impulse transmission part is first from the photoreceptor, because the impulse is always starting from the receptor first. So photoreceptor. So impulse transmission path is first photoreceptor, then bipolar, then ganglion, then to the optic nerve, and then to the occipital lobe of your brain. Okay, the photoreceptor cells are of two types: rod cells and cone cells. So the photoreceptor cells have light sensitive proteins called photopigments. So photoreceptor cells contain photopigments in them. First, let's talk about the cone cells, which are responsible for color vision, daylight or photopic vision. Color vision, daylight or photopic vision. This helps in seeing all the colors around you, whereas the rod cells help in seeing darkness, twilight or scotopic vision. So there are three types of cone cells for each: red, green and blue light respectively. So the combination of all these cone cells help us to perceive the different colors in us around us. So when these cones, all these three types of cones are stimulated equally, we can see a sensation of white light, as white light is a combination of WIB-GR, right? So cones have three photopigments, okay, which is RGB, red, green, blue. And the photopigments in the human eye, whether it's for rods and or cones, it is composed of a protein called opsin and an aldehyde of vitamin A known as retinal. That's why we say carrots, which are rich source of vitamin A, are good for your eyes. So the photopigment is composed of a protein called opsin and an aldehyde called retinal. It's not retinol. So when it's all OL, it is an alcohol. When it is AL, retinal, okay, it's an aldehyde. So be very careful with the spelling given in the option. Don't ever make this mistake of choosing retinol. So next is the rods, which help in twilight or scotopic vision, which is during darkness, because this rod cells contain a purplish red protein called the rhodopsin or visual purple. So cones contain a pigment called visual violet, and rods contain visual purple. Purple R. There is R. So this is for rods. Purple and violet. There's no R. Visual is same for both pigments, right? So since there is R in purple, it is for rods. Remember it that way, you'll never make a mistake. And this visual purple is again contains a derivative of vitamin A. So eat lots of carrot and improve your eyesight. So here you can see the image of ganglion cells, bipolar cells and photoreceptor cells. Alright. These three layers are the nervous part of the retina, which is non-pigmented layers. And there is one more layer called the pigment epithelium, which is the pigmented layer, which is non-nervous. So totally there are four layers of retina. But how many neural layers? Only three layers. Gang of lions surrounded by two polar bears taking a photo. Okay, from inner to outer. Remember that very carefully. So here can you see the central pit? This central pit is called the fovea. Fovea is the thinned out portion of the retina where only the cones are densely packed and rods are absent. And this is the place where the resolution of vision, resolution means visual acuity, that is the sharpness of vision, high resolution we say, right? So this fovea is the place where the resolution is maximum, the greatest. Blind spot is the next topic. Blind spot is a point which is medial and slightly above the posterior pole of the eyeball. Medial, medial and slightly above to the posterior pole. This is very important. So from the very term blind spot, we know that there's no visual activity taking place here, right? So which means both rods and cones are absent. So photoreceptor cells are not present in the blind spot, and this blind spot is the spot where the optic nerves leave the eye and the blood vessels enter the eye. Okay, the nerves leave the eye and the blood vessels enter the eye at the blind spot. So lateral to the eye blind spot is what is present is macula lutea. Macula lutea means, so always what have I told you? Break the word. Macula lutea. So in biology, lutea means yellow. Can you recollect corpus luteum, yellow body, right? Corpus luteum. Similarly, macula lutea. So here macula is a spot. So yellow spot. So macula lutea is a yellowish pigmented spot which is present lateral to the blind spot, and the blind spot is present at the posterior pole of the eyeball, that is a point medial to and slightly above the posterior pole of the eyeball, and the macula lutea is present at the posterior pole of the eye. So carefully notice the difference between the location of the blind spot and the macula lutea with respect to the posterior pole of the eye. So the fovea which we saw is the central pit of the macula lutea. So the last topic left is the mechanism of vision, which is again very important, and you can expect statement based questions like arrange the following in the correct sequence and so on, questions like that. So this is again very important. So how do we see objects? First, light rays from the object, from the visible wavelength, is focused through the cornea and then moves to the lens. Then it is focused on to the retina. The screen, the lens of the human eye is what? You have to find out and tell me. And the screen of the human eye is called the retina. The retina acts as a screen. So when light is focused on the retina, potential or impulse is generated in the photoreceptor cells, which is the rods and the cones. Okay, and this light will induce the dissociation of retinal from opsin. So what is retinal? It is an aldehyde derivative of vitamin A, and opsin is a protein. So light will cause dissociation or splitting of retinal from opsin. So there is a structural change of the opsin, right? Because of the splitting dissociation of retinal from opsin, there is the structure of opsin is changed now. So because of the structure of change in structure of the opsin, the permeability of the membrane changes. So whenever there is a difference or change in the permeability of the membrane, the potential difference is generated in the photoreceptor cells, producing a signal or action potential. So now do you see where is the generation of the potential difference? It is in the photoreceptor, and this travels through the bipolar cells and reaches the ganglion. So I told you initially, right, the path of impulse transmission is from the photoreceptor cells to the bipolar cells, then to the ganglion. It's the reverse pattern compared to the light transmission path. So from the ganglion cells, the action potentials are transmitted by the optic nerve to the visual cortex of the brain. What is the visual cortex of the brain? It is in the occipital lobe. That is at your back side of the head. You can find the occipital lobe of the brain. And what happens in the brain? The neural impulses are analyzed and the image formed is recognized based on our early memory and experience. So repeat with me the mechanism of vision. First, the light rays is focused onto the cornea where the refraction of light happens because it's entering from one medium to another medium. You've learnt it in physics, right? So from cornea it reaches the lens and it is focused on the retina. This generates action potentials or impulses in the photoreceptor cells. Light then induces dissociation of retinal from opsin, causing a structural change in opsin. So this leads to change in membrane permeability, causing potential difference. So the signal is generated first in the photoreceptor cell, followed by the bipolar cells, then in the ganglion cells, and then the action potentials are transmitted by the optic nerves and reach the visual cortex of the brain, where you can where the image is analyzed and recognized. So if you still find this little difficult to understand and difficult to remember the sequence, I have a trick for you which goes like DSP. You know DSP, right? He's a famous musician in the South Indian film industry. So DSP PD. So D for dissociation, S for structural changes in opsin, P for permeability changes, and PD for potential difference, generation of potential difference. And and the first part is clear, right? The light coming and reaching image formation in the retina is quite obvious. So the only confusion will be there in the middle part, which now you can remember through this trick, DSP PD. Okay, here I conclude my video of the eye, and I hope every concept is crystal clear in your mind, and this is an extremely important topic. I am stressing on it again. So I hope I have tried my best by providing many tricks here and there, especially in the confusing parts, for you to remember and recollect in your exam. Okay, thank you. Like, share, comment if this video was useful to you, and please subscribe, as this will motivate me to make better content and provide better short tricks for you people to remember an AS NEET exam. Thank you once again.